It now?
Hello everyone, we are Team 5 and today we are presenting our progress for Senior Project 1. Our team is composed of Amro S Albegirmy, O Farag, Maha M Moussa and myself, Noha A Mohamed, and we are advised by Dr. Mohamed Moustafa. Our presentation will start with a brief introduction followed by the architecture, dataset, signal processing, the models we created, the headset experiments, and outcome and timeline. Okay, so first I will give you a brief introduction to our project. We are implementing a brain-computer interface that classifies EEG signals and converts them into motor commands that can then be used to operate a motor device for individuals with motor disabilities, such as a wheelchair. The motivation behind our project is to improve on the research done in the field of motor classification using EEG signals. The outcome of this project is a research paper that will document our findings and results. All right, so initially the architecture of our project was like the diagram shown here, where we start by choosing and analyzing the dataset. Then we were going to perform some signal processing on the EEG signals in our dataset, and then extract features from them using some feature extraction mechanism that is compatible with EEG signals. We were then going to feed the extracted features to our deep learning model and train it in order to have a classification model that can then read signals from an EEG headset and classify them into one of 4 different classes that are then translated into motor commands. However, through our research, we have seen that the feature extraction unit can be separated from the design, and we could have an architecture where our deep learning model does the feature extraction for us, and we will describe that later on in the presentation. Also, we could include the feature extraction unit for better classification results from the model. These are the 2 different designs that we followed in our implementation. Before we begin talking about our model, I will first discuss with you the dataset we chose. Through our research, we discovered that the Physiognomy dataset is the best one for our purposes, and it has been used by other researchers and produced good results, so we decided to go with the Physiognomy dataset. The sampling rate for all signals is 160 Hz, which is sufficient for an EEG signal. The dataset has 109 subjects, which is good because it allows our model to be more generic. There's approximately 1,500 EEG recordings, and these recordings belong to 5 different classes. Each one of the 109 subjects were asked to perform 14 trials. Each trial lasts for 125 seconds. 2 out of the 14 are base runs to identify the baseline signals. The odd number trials are actual movements of left or right fists, or both fists or both feet, while the even number trials are imagined movements. The rest state is given the label T0, while the labels T1 and T2 can actually be divided into 2 other labels based on which run it is. So T1 would just indicate left fist, T2 would indicate right fist, T3 would indicate both fists, and T4 would indicate both feet. Since an event occurs in the EEG signals for a period of time, the signal is labeled by dividing the signal into chunks containing the beginning of each event. We can differentiate between different events because they are separated by the rest event. For instance, in the array shown here, we can have an event that starts at 0.672 and it lasts until the next rest event, which has a label 0. So we can see that all the events are separated by the rest event, which can be seen from the occurring zeros. Okay, so now I will discuss the feature extraction methods that we chose from. The most widely used feature extraction methods in the field of EEG signal classification are the independent component analysis, which reduces a large multivariate signal into smaller additive components. There's also the short-term Fourier transform, which provides the time-localized frequency information for situations in which frequency components of a signal vary over time. There's the autoregression model, which forecasts future behavior in a signal based on preceding input. The wavelet transform transforms time domain signals into time frequency domain signals. And the last method is the common spatial pattern, or CSP, and it is the method we actually chose to proceed with in our first approach towards training our data. All right, now we will talk about the different approaches we are following to train our chosen dataset. The first approach we took is based on the CSP feature extraction method that I mentioned earlier. We wanted to assess the performance of our model when we apply the different feature extraction methods to the model. We did a lot of research on the best feature extraction method for our data and our objective. And what we found is that most papers who attempted to achieve similar objectives as ours went with CSP, and it produced the best results. Therefore, we decided to first try CSP with a neural network and measure the performance of the produced model. CSP is used in signal processing for separating a multivariate signal into additive subcomponents, which have maximum differences in variance. CSP transforms EEG signals to a new space, maximizing the variance of one class while minimizing the variance of the others, making them more distinguishable. If we assume we have the data of two classes, like the motor-imagery of right and left fists, CSP calculates filters that maximize the ratio of variance of data stemming from the two classes. CSP allows for information to be extracted from different frequency bands, and since EEG signals have different information in various bands, then CSP is suitable for use with EEG signals. CSP is used in signal processing for separating multivariate signals into additive subcomponents, which have maximum differences in variance. CSP transforms EEG signals to a new space, maximizing the variance of one class while minimizing the variance of the others, making them more distinguishable. If we assume we have data of two classes, like motor imagery of right and left fists, CSP calculates filters that maximize the ratio of variance of data stemming from the two classes. CSP allows for information to be extracted from different frequency bands, and since EEG signals have different information in various bands, then CSP is suitable for use with EEG signals.
Second approach we used involved the conversion of EEG signals into images and using a recurrent convolution network for training. The aim of this approach is to process the EEG signals in a way that will preserve their spatial, spectral, and temporal structure. Most of the research papers that we've looked at use the convolutional neural networks together with the recurrent neural networks for the training of the EEG signals. The only difference was in the architecture, i.e. whether the two networks were cascadal or parallel, and the number of the layers of each network. The first step is to convert the EEG frames into images to preserve the spatial structure. The images must have multiple color channels to represent the spectral dimension. The sequence of images produced from multiple consecutive time windows accounts for the temporal changes in the brain activity. In order to be able to represent the readings of the electrodes, which are originally distributed in a 3D space on a 2D surface, we use the azimuthal equidistant projection. This way, the distance between the electrodes and the center point is preserved. The width and height of the images represent the spatial distribution of the activities over the cortex. The next step is to use the Kloff-Tucker scheme to interpolate the scattered power measurements over the skull and interpolate the values in between the electrodes over the 32 by 32 measure. This procedure is repeated for each of the frequency bands of interest, resulting in multiple unicolor topographical activity maps. These topographical maps are then merged together to form a 3-channel image. And as you can see in the image below, the first graph represents the location of the electrodes in the original 3D space. The second part represents the projection of electrode location using the topology preserving azimuthal projection. And the last part represents an image after the Kloff-Tucker interpolation was applied. The components of the network are CNN, LSTM, 1D convolutional layer, fully connected layer, and a softmax layer. The images are used as inputs to a 7-layer parallel convolutional network with shared weights, and the output is fed to the 1D convolutional layer and the LSTM. Then the output of those acts as an input to the fully connected layer and the softmax layer for classification and loss function calculation. Convolutional networks were used to deal with the variations in space and frequency domains due to their ability to learn good two-dimensional representation of data. LSTM was used to capture temporal changes in the activation of the convolutional neural networks. Since brain activity is a temporally dynamic process, variations between frames may contain additional information about the underlying mental state. 1D convolution is applied to the convolutional network output to capture any temporal patterns across multiple frames. This is a diagram that clarifies the whole process. EEG time series from multiple electrode locations are required. These are used to form a sequence of topographical maps as I mentioned before. And these topographical maps are used to form a sequence of images that are fed to the convolutional neural network for spectral and spatial feature extraction. The convolutional network output is fed to the recurrent network for temporal features extraction, and finally, we got the class prediction. We're currently still working on this model as we want to see the accuracy and the result it will produce on the dataset. However, till now, we believe that the models A and Amro will explain next are more efficient as they don't involve the pre-processing and the conversion of EEG signals into images.
Okay, our third approach is the cascade and parallel CNN LSTM. This approach requires no signal processing and only the normalization. So our goal is to implement a code which excludes pre-processing as much as possible. And since the headset has 3D locations over its skull, it's essential for the model to learn from spatial patterns and temporal patterns. Therefore, after obtaining the data the same way we described before, the data is then converted into a 2D mesh that represents the locations of the electrode so that the stacked CNN can grasp the spatial pattern. So what we're doing is building a mesh-like EEG hierarchy from our 1D EEG chain-like vector by mapping the EEG recordings with spatial information of the EEG electrode in order to correlate between neighboring electrodes and corresponding brain areas. So now when we look at the cascade CNN RNN, here we have the input to the model is a 2D mesh, and then the model creates a 3D data architecture. So what it does, it first extracts the spatial features, and then it feeds it to the RNN model to extract temporal features. And the output of the RNN is then fed to the softmax layer for final prediction. And then we look at the parallel. Here, in the parallel, it's exactly the same as the cascade, but it differs in the fact that both models are extracting features in parallel rather than after each other. And when we look at how we convert the 1D EEG sequence to a 2D EEG mesh, so here the subject imagines or performs an action, and the electrodes then acquire the fluctuations of the voltage from the skull. The readings from the EEG represent a time series data, and it's a one-dimensional vector as shown here. So after that, from the EEG electrode map, here we can see each electrode is physically neighboring multiple electrodes in a certain area, and each area corresponds to a different brain activity. So while the 1D EEG vector only is restricted to two neighbors, the one above it and the one below it. So for this reason, we convert the 1D EEG data to a 2D mesh in order to try to reach this conceptualization of the EEG electrode map. So, and as you can see here, all the zeros here represent a null electrode, which means they have no effect in neural network. And after that, the data, the 1D data vector series that have been turned into a 2D mesh here is normalized using a Z-score normalization. And after that, we apply the sliding window in order to divide the 2D mesh into clips with 50% overlapping between neighbors.
Implemented the cascade CNN-RNN model. The 64-channel signal is converted to a 2D mesh where each mesh appears in this slide as a slanted box. This model uses the idea of a sliding window where the window moves and reads only the meshes inside the window at a time. Then it produces a prediction based on spatial and temporal features. When the window moves, it has a 50% overlap with the previous input as seen in this slide. The first frame in each of the slice signal goes to the first CNN. The second frame in each of the slice signal goes to the second CNN, and so on. We end up with number of CNNs equal to the number of frames in the window cells. We have tested different window sizes, and our best result had a window size of 40 frames, where all CNNs had the same architecture. That is, it receives a single frame or a single mesh of 10 by 11 by 1, and it goes through 3 convolutional layers. All has a filter area of 3 by 3, but with depth of 32, 64, and 128 respectively. Then there is a 1024 neuron fully connected layer, and finally a 5 neuron layer which produces a loss using cross entropy and self-mix. Please note that there is no pooling layers used. And after training the 40 CNNs, the last 5 neuron layers was chopped off or removed, and the output from the 1024 neuron layer is taken as an input in the LSTM. The second part of the model is to train an LSTM neural network. We have used the frame from each window as an input to the saved CNN models to produce its 1024 neurons. Then each of the 1024 neurons is an input to an LSTM unit that has 64 hidden units. There are 2 layers of LSTM, such that the second layer's last unit output is 5 neurons, which is used to make the prediction and calculate the loss using cross entropy and softmax. This LSTM is used to extract the temporal features after the spatial features has been extracted by the convolutional neural network. After implementing this model with different window sizes, we have found that the best window size was 40 frames, which produced a validation accuracy of 91.5%. In order to summarize the models that we have implemented, we have done pre-processing experiments, and we have found that they were the most advanced and has produced moderate accuracy results that would not be able to challenge the existing research papers. The image-based experiments are still being developed, and we have not produced any results for those experiments yet. The cascade CNN RNN experiments were the least complicated and gave the most promising results. As for the future experiments, we are planning to improve on the cascade CNN and RNN, such that it will act as the starting point for our experiments. Therefore, we will start by combining slide preprocessing like bandpass filter with the cascade CNN RNN. We will also then, for experimentation purposes, try to combine advanced preprocessing with the cascade CNN RNN. And as soon as the image-based experiment results are out, we'll evaluate its performance and see if it's worth combining also as well with the CNN RNN approach. For each of those experiments, we would like to analyze the model performance in different subjects other than the ones being trained. And then we will also try to evaluate the model accuracy as well as its speed.
Right now, we'll be discussing the headset. So we're currently using the OpenBCI headset, the one shown in the picture here, and it has 8 electrodes, and we've been working a lot with it, testing and collecting data. We're collecting data the same way that the PhysUnet dataset collected its data. For each subject, we record 14 different runs, similar to the dataset's experimental runs, which include 2 baseline runs, tasks, eyes open and eyes closed, and then 4 different tasks, which have open and closed left or right fist, and then imagining opening and closing left or right fist, then open and close both fists, and then task 4 is imagine opening and closing both fists. And we do this 3 different times. So we collected data from 2 different subjects and noticed an apparent issue in the signal, which is the following. The electrodes just provide noise and nothing else, and the electrodes that are on the forehead are the only electrodes that actually read a proper signal. And we can see from it eye movements, and the rest of the electrodes just provide noise and nothing else. And this is a major issue as shown in the video. Okay, so the eye movements can be seen from the second electrode here, so which is right here. So here the subject blinks once, here the subject blinks once, the subject blinks twice, and here I ask the subject to blink multiple times, and then ask the subject again to blink once. And so you can see that only the first one shows an actual signal, and the rest we can't read anything from it, and it's actual noise, and so does the first one. So this is an apparent issue. Another issue, it takes us a very long time to start reading a proper signal, and to read a proper signal, it can read at random times. And for us to read different runs, we must reset it every single time, which is completely inefficient since we actually use it for disability. If we actually use it for the disability community, taking a long time in order to get the headset to read an actual signal is an issue. And it is actually extremely painful to wear, as the electrodes have pins and you push them on the scalp. And since we're using dry electrodes also, they provide a lot of noise, so this is not helping at all with the signal. Therefore, after a lot of considerations, we ordered another headset from Emotiv. And why is it better? Why Emotiv? The electrodes are not dry. It has 14 electrodes rather than 8. The setup time is 3 to 5 minutes. It's flexible, and the electrodes don't have pins in them. So it's actually it's better to wear them, and it's more efficient to have. So, and we're expecting it by the end of December. So, and our plan is when we receive it, is we will be collecting data again, and we're aiming for 15 to 20 subjects this time, and we'll be adding the data collected to our training data set. Also, we'll be editing our data set in order to match the headset. As for our outcome, our main outcome in our project is a research paper. So these are the steps in general to publish a research paper, a scientific research paper. So the first step is select a topic, which we've done, identify the problem within the field, which we have, collect data and analyze the data. We're working on both, and analyze the data focuses on the idea of here, our way of analyzing data is having a model. Then our last two steps focus on writing the research paper and publish the paper, and we're still there. We still haven't reached this point, but as you can see from our presentation so far, we've been working on these 4 steps.
This was our timeline for Thesis 1. I'm glad to say that we're on track as we are now in the phase of model training. We will need to work on the signal processing part again in Thesis 2, as we believe it can help us get better accuracies for our model, and we will need it for the signals obtained from the headset. As for Thesis 2, we'll be mainly working on improving the model, and we expect that to be done by mid-March. We're also waiting for the new headset to arrive so that we can start using it to collect data and add the data to the training dataset. Then we can start testing our model with the signals obtained from the headset. We expect to finish the testing by the beginning of May.